Hello and good morning students. I am SN Oja, PGT Bio from KV number 1 Riva. Students, you all have seen the series of video lectures on your biology topic. Today we are going to discuss a very important concept of molecular biology. As in the previous chapter, you all have studied that genetic materials are the biochemicals, biomolecules which are inheriting from one to another generation. We have also studied about the few important characteristics of a genetic material, but then the question arises how we came to know that what is the actual genetic material, and second question, that what is the structure of this genetic material. From today we are going to start the chapter that is molecular basis of inheritance, a very important chapter. And in this chapter for today we are going to study about the chemical structure of the nucleotide and how this genetic material are composed of. So let's begin our topic. Well, today we are going to discuss only one topic of this whole chapter, that is the molecular structure of nucleotide. In this topic, we will study about the structure of DNA, the nucleotidic composition of DNA and the biochemistry of DNA. At the end of this chapter, we will also study about the Watson and Crick double helix model. You all have a little bit idea since you all have studied in your previous class 11th, but we will highlight it off and we will also discuss the Chagaff rule and how it will be helpful. Okay. Well, talking about the genetic material, previously there was a series of experiment, and you all know about that there was a series of experiment, and then only today we come to the conclusion that DNA, that is deoxyribonucleic acid, is the genetic material. Fisher has first of all isolated this, and it has termed this genetic material as nuclein, since it is present inside the nucleus. Fisher has also postulated its acidic role and acidic behavior of this biomacromolecule. In class 11th, if you remember, various types of macromolecules we have studied, and the largest molecule are DNA or nucleotide. Okay. Well, if we're talking about the DNA, deoxyribonucleic acid, that is also called as DNA, and ribonucleic acid, that is also called as RNA. These are the two type of nucleic acids. So again, let me repeat or emphasize my word, that is nucleic, and they are acid. They are present inside, they are nucleic origin, and they have a acidic property, which are found in the living system. So variety of the living systems are there, right from, if we talk about virus, bacteria, other prokaryotes, eukaryotic, unicellular, eukaryotic, multicellular, and what that complete living kingdom, all this living kingdom has the genetic material, that is DNA or RNA. DNA is act as a genetic material in most of the organism. Again, a very interesting question. When you study this whole chapter, slowly you will came to know that RNA is more important than DNA. But still DNA is the genetic material in most of the organism. So again the question rises, when RNA is so important, why DNA has superseded the RNA in the due course of evolution? So that also we will see in some other videos. RNA, though it is also acting as a genetic material, but it is acting as a genetic material mostly in viruses. Viruses, the structure of virus, you all have studied in class 11. So RNA is acting as a genetic material, but only in the viruses, and in most of the eukaryote, they are acting as messengers. They are carrying out the messages from nucleus to other system for the protein coding. So if you talk about the messaging system of this, if you talk about the messaging system, how it all happened, that it is DNA in eukaryotes. This DNA codes for RNA, also famously called as mRNA, and this RNA again codes for protein. So here the RNA, mRNA, messenger RNA, rRNA, ribosomal RNA, and tRNA, transfer RNA, all the three families of the RNA is responsible for coding a protein, and that's why our protein based upon the genetic material, the type of genetic material what we all have. Okay, so here it is acting as a messenger. Well, RNA has an additional role as well. It functions as adapter molecule. Now only I was discussing about adapter molecule. Now only I was discussing about tRNA, that is a transfer RNA. So in the due course of this chapter, that also you will study that this tRNA or the adapter RNA, they carry we all know that the proteins are made up of amino acids. So this tRNA carry one amino acid and pair it to the another amino acid. That's why they are called as an adapter RNA. They are also playing a very important role in the structures. For example, ribosomes. Ribosomes are chiefly made up of ribosomal RNA, also known as rRNA, and in some cases they are acting as catalytic molecules also, which initiates various types of catalytic reactions. So this is a little bit of overlook about DNA and RNA. Well, if we talk about nucleotide, so first of all we should know what is nucleotide and what is nucleoside. Nucleotide and nucleoside you all have studied in class 11th. Nucleosides are the sugar plus nitrogenous base. So sugar, any pentose sugar or sugar plus nitrogenous base, it is termed as nucleoside. And when a phosphorus group is added in this, it is termed as nucleotide. So nucleotide is a combination of three things: sugar, nitrogenous base and phosphate group. So that nucleotide, what are the nucleotides which are available in the living kingdom and their function, how these nucleotides are playing or behaving as an important function. So the first and very important role of a nucleotide is nucleotides are the building block of DNA and RNA. If you see the biochemical structure of DNA and RNA, it is made up of various types of nucleotide: adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, uracil triphosphate, thymosine triphosphate. So these are all the nucleotides which are acting as a building block for the biomacromolecule, that is DNA and RNA. Another, they are acting as the intracellular source of energy, the most important biochemical source of energy, and in which the energy is stored in our body, that is called as adenosine triphosphate. This adenosine triphosphate itself is acting as a nucleotide. Next one, they are acting as a secondary messengers. In class 11th, we have studied about hormone action. In that hormone action, if you see, you have seen this word cyclic AMP, cyclic adenosine monophosphate. This cyclic adenosine monophosphate it acting as a messenger, secondary messenger. Actually what happened, for example, this is my plasma membrane, okay, and on this plasma membrane we have few receptors. On this receptor, whenever any hormone attached, this is suppose the hormone, so hormone attached on the receptors, right, then here a cascade mechanism involves. So here hormones are acting as a primary messenger because they are the prime source, they are the prime biomolecules which carry the messages, and then it comes to it attached to the plasma membrane, and from the plasma membrane it again carry forward various types of secondary, it generates secondary messengers, and that secondary messengers undergoes a cascade mechanism, and that cascade mechanism is responsible for amplification of any signal, and that's why eukaryotic body promptly or enormously responds for any external signals, right. So here it generates cyclic AMP, cyclic adenine monophosphate. So that is again also a very important function of nucleotides. The next important function, they are acting as an intracellular signals. For example, intracellular signals means cell to cell. There is a well proper cell to cell signaling. One cell can signal its message to the another cell, and this signal is nothing but due to the G proteins. So G proteins are acting as an intracellular signaling. They are very important for the intracellular signaling. So this is all about the few nucleotides and their few specific functions. Next is, let's begin today's topic, and that is the DNA. Why I am emphasizing on this molecule? A very interesting, very important molecule, DNA. It is the molecule which in the course of heredity we all know it is a molecule which control almost all the biochemical functions of the body, and the core molecule which is responsible for evolution, right. So evolution can only takes place when there is a change in the structure of a genetics, because this DNA control the characters. And how it control the character? DNA control the character through proteins, and DNA codes for the proteins, and proteins control the character. So when DNA changes, proteins also changes. When the sequence of proteins or sequence of amino acid changes, characters also changes, and that is the main thing behind the diversity. Okay, DNA, the chemical composition of DNA is same, whether you talk about a plant or a bacteria or a human or animals, all have same chemical compositions, but the sequence of these nucleotides are different, which is going to play a very important role in the course of evolution. Okay, DNA, deoxyribonucleic acid. Well, it is a long polymer of deoxy. Again I am emphasizing on this word. Why? We will discuss. Deoxyribonucleotide, right. The length of the DNA is usually defined as the number of nucleotides. So how long is the DNA? We cannot measure it into the kilometers or meters. As in the kilometer, meter and centimeter we are measuring the distance, similarly a length of the DNA can usually be defined as the number of nucleotides or base pairs. We used to say it as base pairs, BP, base pairs, or kilo base pairs, KBP, kilo base pairs. Okay. DNA, as I told you, it decides the characteristic of any organism. So because DNA codes for the protein and protein codes for the character, so that's why DNA ultimately decides for the protein. Well, the length of DNA varies in the due course of diversity. Okay, DNA are very small also. Very small means don't think I am talking about centimeter or millimeters. They are too small, right? And DNA is of very large also. So here just there is few data which is given in your NCERT book about the length of the DNA. Let me take it. Phi 174. Phi 174, it is a very simple virus, right? And this virus has only 5,386 nucleotide, very less number. If we talk about bacteriophages, and it is bacteriophage lambda. Bacteriophage we all know, that is the viruses which attacks on the bacteria. Okay, this bacteriophage lambda has 48,502 base pairs. E. coli, a very common bacteria we have studied in a lot of time. E. coli has 4.6 into 10 raised to power 6 base pairs, right? And in a haploid content of human DNA, it is 3.3 into 10 raised to power 9 base pairs. 3.3 into 10 raised to power 9 base pairs for the haploid set. So you can see here that if you talk about the complexity of an organism or complexity of a body of an organism, the complexity of a body of an organism changes and the length of the DNA is also keep on changing. If you talk about the complexity, viruses are less complex, only they have few amount of nucleotides. While talking about bacteria, they are more complex than viruses, their DNA length increases, and human beings, a highly complex organism, the length of the DNA you can see here, that is 3.3 into 10 to the power 9 base pairs. Just a comparative to understand the length of DNA. The length of DNA, as I told you, it changes from one to another organism, right? Well, talking about the structure of this polynucleotide chain, the length of DNA we all know, the structure of polynucleotide chain is again important. Basically, there are three things which makes this structure, okay, and they are, the first important one is a pentose sugar. A pentose sugar is very important. Why? Because this pentose sugar is the building block. It is the building block for the DNA. Second one is a nitrogenous base. Pentose sugar having 5 carbon in their chain, and nitrogenous base, that is adenine, guanine, cytosine, uracil, and whatsoever it is. A third important point which is not present in nucleosides but only present in nucleotide, that is phosphate group. So these three important building blocks or compositions are very important for deciding the chain of a polynucleotide, that is chain of a DNA. So by the time we have seen that three important components of a nucleotides are there. Sugar, that is a pentose sugar. Second one is nitrogenous base, and third very important is phosphate group. Now let us emphasize and focus on these structures a little bit more. Well, starting from the first part, that is sugar. Nucleotidic structure has pentose sugar. Sugar can be triose, tetrose, pentose, hexose, and further on. For example, glucose is a hexose sugar because 6 carbons are present and 5 carbon are present in the ring. Similarly, when we are talking about DNA has pentose sugar, and that means 5 carbons are present in the ring. These 5 carbons are termed in or numbered in a cyclic way, or clockwise I should say. For example, this is the oxygen and these are the carbons. These carbons are termed or numbered as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. This carbon is first prime, 1 prime, second prime, third prime, 4 prime, and 5 prime. On 5 prime carbon, there is a hydroxyl group. So this is the skeletal structure of the pentose sugar, and this pentose sugar is of two types in nucleotide. One is termed as ribose and another one is termed as deoxyribose. Well, just you compare the structure of this is ribose, this is deoxyribose, and if you compare the chemical structure of these two, we will see some minute differences. For example, in ribose sugar, this is 1 prime, this is 2 prime. On second prime carbon, you will find here a hydroxyl group. That's why it is termed as ribose. But similarly, if we talk about deoxyribose sugar, in deoxyribose sugar, this is 1 prime and this is 2 prime. At 2 prime position, you will only get a hydrogen, but not a hydroxyl group. This is a major difference between ribose and deoxyribose, and that's why this is called as deoxy, D, absence, oxy, oxygen. So one oxygen in deoxyribose sugar is less than what we have in ribose sugar. Ribose sugar are present in RNA, they composes RNA, while deoxyribose sugar, they are present in DNA. And that's why RNA is called as ribonucleic acid and DNA is called as deoxyribonucleic acid. So I think I have made myself very clear about the structure, the difference of the two sugars which are present in our nucleic acid. Well, now let us talk about nitrogenous base. Nitrogenous base are broadly classified. They are the nitrogen containing bases in which their heterocyclic ring has nitrogen. These nitrogenous bases are basically categorized as purine and pyrimidine. That is purine and pyrimidine. There are two types of purine, that is adenine and guanine, and there are three types of pyrimidine, that is cytosine, uracil and thymine. If you see the structure of these purine and purine are bicyclic compounds, so they have two rings, and pyrimidines are monocyclic compound, that means they have only one ring in their structural composition. Okay. This purine, adenine and guanine, they are present in DNA as well as in RNA. But if we talk about pyrimidines, cytosine is present in both. The cytosine is present in both DNA as well as in RNA. But if we talk about uracil, uracil is a pyrimidine which is only present in RNA and absent in DNA. Similarly, if we talk about thymine, thymine is a pyrimidine which is only present in DNA and not present in RNA. So uracil present in RNA, thymine present in DNA. Okay, so that's why it is written here that cytosine is common, but uracil is present in RNA in place of thymine, and this molecular structure, this change also impart the extra stability to the DNA. We will see how. Well, this is what is the skeletal structure of purine and the types of purine. So first of all, let me talk about the skeletal structure. As I told you, purine are bicyclic compounds. So there are two rings, as you can see here. This is the first ring and this one is the second ring. This is the first ring, don't confuse, this is not the first ring. Okay, because in heterocyclic compounds, whenever we are naming anything, that name should be clockwise. Okay, so here there are two rings and you can see the place of nitrogen. So nitrogen is placed at first position, third position, 7th position and 9th position. Due to this, they are termed as nitrogenous base, because aminic group in chemistry you all have studied, amines are basic in nature. That's why they are termed as nitrogenous base. Well, talking about two things, that is adenine and guanine, there are two types of purines, adenine and guanine, and they both have same cyclic compound. You can compare the position of nitrogen. The nitrogen is placed at the ring nitrogen is placed at the same place. Here also you can see, here also you can see. This also you can compare, this also you can compare. At 7th position also you can compare, 7th position also, at 9th position, at 9th position. So the skeletal structure of adenine and guanine both are same. What makes the difference? The difference is, at this position you will find an amine and at this position you will find a ketonic group. Okay, this ketonic group provide extra stability to guanine. That's why in later part of this chapter, we will study that polyguanization is important for making a DNA stabulated. Okay, how? So that all we will discuss here. And in guanine, one amine group, the amine group which is placed here it is actually ketone, and the amine is shifted to this position. The amine is shifted to this position. So I think the structure of both of this is very important, though nobody is going to ask you the structure in the examination, board examination, but it is my personal suggestion, if you want to crack NEET or any competitive examination, be careful with your biomolecules, because the reasoning questions can be drafted on the basis of this, right? So this is about the purine, adenine and guanine. Similarly, if we talk about pyrimidines, so pyrimidines are monocyclic in nature. In pyrimidine, you can see here that there are only two nitrogen. One is present at 1' and another one is at 5th position, right? And this skeletal structure is same whether it is thymine, whether it is cytosine, or whether it is uracil. Thymine is present in DNA. It is not present in RNA, as I have already told you. This thymine, if you compare both of this structure again, in thymine there is an extra ketonic group, and in cytosine, instead of that, there is an amine group, and this here the ketone group is same. At this position you can find a ketonic group. Here also there is a ketonic group, and in thymine there is an extra important component, that is methyl. This methyl play a very important role in again maintaining the stability of the DNA. Okay, so that are the two structures of thymine and cytosine. Well, now I would like to compare the structure of thymine and uracil, because they both are pyrimidine, both are monocyclic. Thymine is present in DNA, uracil is present in RNA, and you also know that DNA is a stable molecule as compared to RNA. One of a very good reason is the availability of thymine in the DNA. Okay, let us discuss how. If you compare the structure of thymine and uracil, you will not find any change. All are same. Thymine, ketonic group, aminic group, ketone, amine. Just only there is one change, and that is this is first, this is second, this is third. At 3' position, here there is a methyl group, and at 3' position, here nothing is present. Okay, only carbon is present. So this methyl group imparts the extra stability of this thymine molecule. Right, that's why it is important that that's why the DNA is more stable as compared to RNA, because DNA has thymine and RNA has only uracil. Right. So now I think I have made myself little bit more clear. DNA, the polynucleotides, how the structures are, and what are the stability. These points, keep it in your mind. When we come to the last of this topic, maybe in some other video, we will compare the structure of DNA and RNA, and that time I will tell you what is the basic reason. So by the time, two important differences we have studied. If you talk about sugar, RNA has ribose sugar, DNA has deoxyribose sugar. If you talk about nitrogenous base, RNA has uracil, which is non-methylated, and DNA has thymine, which is methylated. Okay, well, now this is about the nitrogen. So two things we have discussed, that is sugar and nitrogenous base. The third important point is the phosphate groups. Phosphate groups are the derivative of phosphoric acid, and these phosphoric acids are important for making bond. Okay, and that is most importantly or basically termed as a backbone of DNA, also called as phosphodiester bond. Right. So this is the basic structure of a phosphate group. That is, as I told you, a phosphorus is there, oxygen, and here whatever the group which is going to attach. This phosphate group is very important in polymerization. polymerization of nucleotide. One nucleotide should connect to the another, should connect to the another, should connect to the another. So for example, अगर हम लोग घर बना रहे हैं, ईंट तो हमारे पास है, तो ईंट क्या हो गया, पेंटोज शुगर हो गया, नाइट्रोजनस बेस हो गया. सीमेंट का काम कौन करेगा? सीमेंट का काम ये फॉस्फेट ग्रुप करेगा, तभी जाके वो पूरा का पूरा ब्लॉक जो है वो बन पाएगा. In the same way, here also there is this phosphate acting as a linking part between one brick of nucleotide to the another brick of nucleotide. Okay. Well, if we talk about more little bit about the phosphate, so phosphates can be the nucleotide can be monophosphate, it can be diphosphate, or it can be triphosphate. While talking about the monophosphate means whenever we are talking about, for example, I have taken an example of AMP, that is adenosine monophosphate. Here there is a adenosine, this is the carbon of adenosine, okay, and this adenosine is connected with a single phosphate group. That's why it is called as adenosine monophosphate. If two phosphorus groups are there, that is called as diphosphate. Famously we all know about ADP, adenosine diphosphate. Again, this is the sugar molecule, this is the carbon of a sugar molecule, and you will find one phosphorus here, another phosphorus here, that is 2 phosphorus. Third, if we talk about adenosine triphosphate, a very famous molecule of energy. So here you can see that this is the carbon of a sugar, pentose sugar. This pentose sugar connected to one phosphorus, another phosphorus, and third phosphorus. 1, 2, and 3. That's why it is called as adenosine triphosphate. Okay. So in the same way, in cyclic AMP we will find a cyclic type of molecule which is connected to the phosphorus, and GTP guanosine triphosphate, or CTP cytosine triphosphate, or whatever is the molecule, they are connected with the phosphorus and they are called as monophosphate, diphosphate, or triphosphate. Well, now very important part here is the linking. We have discussed about the sections, we have discussed about the different sections. We have discussed about part 1 of nucleotides, we have discussed about phosphate bonds, and now we are going to discuss about the various types of bonds which are present in the DNA. Basically, in the DNA, 2 nucleotides, they are linked by 3', 5' phosphodiester bond. 3', 5' phosphodiester linkage. Okay, what is that? Now we will discuss. More nucleotides can be joined in such a manner, and as a result this is called as polynucleotide. One nucleotide, another nucleotide, another nucleotide, another nucleotide, and then it is termed as a polynucleotide, right? A polymer thus formed at one end free phosphorus. So whatever a long polymer which is formed, this long polymer has 2 ends. At one end the phosphate group is free, and at another end the hydroxyl group is free, and as a result this provides the polarity of the DNA, and hence DNA is termed as 3' to 5' or 5' to 3'. The strands of DNA are termed in that way. Okay, so there is a 5', a phosphate is present at 5' end, and hydroxyl group is present at 3' end. Okay, the backbone of the DNA is nothing but it is the phosphodiester bond, which is composed of sugar and phosphorus, and the nitrogenous bases are connected together. One nitrogenous base of one strand is connected to the another nitrogenous base of another strand through hydrogen bonds. Okay, so this is simple an introduction I just want to give here about the bonds, and now this diagram is very important. In the molecule of DNA, if we discuss, there are three different type of bonds which are present. The first bond is called as phosphodiester bond. Phosphodiester bond. These phosphodiester bonds are acting as a backbone of the DNA. The second bond is called as glycosidic bond, which are responsible for linking of a nitrogenous base to a sugar molecule. And third bond, that is called as hydrogen bond. Hydrogen bond, which connect two different strands of a DNA. Right. Let us discuss from this diagram one by one. First one, as I told you, that is phosphodiester bond. So if you see here, this is a sugar. At this sugar, here it is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. This is another sugar molecule, 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. So here there are two sugar molecules. Let me draw it here. One is like this, another one is like this. I am discussing the first type of bond, that is phosphodiester bond. So here what happened, there is a hydroxyl group, OH, and here there is a carbon with an OH, and here phosphate is present. A bond is established between this oxygen and this phosphorus, which is shown here, if you can see this. Similarly, we can see also here. So a bond is established between one sugar to another sugar and between these two molecules. This is termed as phosphodiester bond. This is termed as phosphodiester bond. Here also it is termed as phosphodiester bonds. So these bonds are acting as a backbone of DNA. And one more thing, here you can see what I was discussing, at 5 prime end one phosphate is added. So phosphorus groups are added only at the 5 prime end. Okay, and this at this 5 prime carbon you will find here there is a phosphate group, and here at this is a 3 prime end, so here it is a PO4 3 minus, a group is there, and here there is a group wherever group. So wherever in the nucleotide hydroxyl groups are present, that will denote 3 prime end. Wherever 5 prime ends are there, that will be termed as a phosphate group. That will be termed as a phosphate group. So this is the first type of bond, that is called as phosphodiester bond. Second type of bond, that is the glycosidic bond. When you are discussing about glycosidic bond, it is also called as N-glycosidic bond. Why called as N-glycosidic bond? Because it is the linking bond between the sugar and the nitrogenous base. So for example, if this is my sugar, this is the first carbon. From first carbon, here the nitrogenous base is added. And this bond, through which a nitrogenous base attached to the sugar, this is termed as glycosidic bond. Glycosidic bond, or N-glycosidic bond, because one nitrogen involves. As you can see here, has the guanine is attached. So this guanine has 1 prime, this is the first prime carbon, and this has a bond with the nitrogen. So that's why it is first 1 prime nitrogen. Okay, so these are the two bonds we have discussed. The third bond, that is hydrogen bond, that we will discuss when we discuss about the helix of the DNA. Right. So again, it is the sugar and phosphate bonds, as I had already discussed here. The third one is the nucleotides. These are the nucleotides, and then they are bonded with each other, right? Well, base pairing is very important in the structure of DNA. Irwin Chagaff, he has proposed the base pairing rule. So that we will discuss after the Watson and Crick model. But here, simply we are discussing now the biochemistry, and today is the first lecture. So just I want to highlight some important parts of that. So here what we will see, that these two strands, there is one strand of one DNA, there is another strand of another DNA. These two strands are anti-parallel to each other. One strand run from 3' to 5', and another strand run which is parallel to this, that is from 3' to 5'. So both strands are connected together. But in RNA, if we talk, there is only one strand. So in the RNA, since RNA is a single stranded polymer, so that's why no further things are there. But if we're talking about the DNA, DNA is a double stranded polymer. So double stranded polymer means, for example, this is the one strand and this is another strand, and both these strands are connected. Here also some nitrogenous bases are there, here also some nitrogenous bases are there, here also nitrogenous base, here also, here also, here also, here also. So these nitrogenous bases are connected through a weak van der Waals forces or weak interaction, and there due to the hydrogen bonds. There are two hydrogen bonds. Adenine always pairs with thymine. This is the base pairing rule given by Irwin Chagaff. Adenine always pairs with thymine and guanine always pairs with cytosine. Adenine of one strand. Now you can understand it like that. Suppose this is one strand and suppose this is another strand. So adenine of one strand connected to thymine of another strand by two hydrogen bonds. At the same time, guanine of one strand connected to cytosine of another strand again by 3 hydrogen bonds. So G pairs with C by 3 hydrogen bonds and A pairs with T only by 2 hydrogen bonds. It is again a 1 mark question. It should be important to note. Okay, this is how you can understand that how 2 and 3 hydrogen bonds are connected. So here suppose this is thymine. This is the thymine and this is the adenine. So T always pairs with A by 2 hydrogen bonds, and these are the 2 hydrogen bonds. And this is the cytosine, here it is guanine. C pairs with G by 3 hydrogen bonds. So here it is the 3 hydrogen bonds. Okay, this is one strand, this is another strand. The one strand running 3' to 5'. The another strand, the polarity of another strand is 5' to 3'. So both the strands are anti-parallel and both the strands are connected with a particular type of hydrogen bonds. So if you talk about the RNA, usually RNA is a single stranded structure, so that's why no hydrogen bonds are present. But in some cases, for example in tRNA, in transfer RNA, when the RNA is coiled, the two strands come closer to each other. Okay, and there also partial hydrogen bonds are established, right? Now, so this is how our DNA appears, the two strands. Okay, this is suppose this is 3' end, this is 5' end, here it is 5' end, here it will be 3' end. Okay, this DNA remains in the double helix structure. We all know about the Watson and Crick model. That also we will discuss in my next video lecture about the Watson and Crick model and Irwin Chagaff rules. Right, and if we talk about the 3D structure or 3-dimensional structure, the DNA something appears like that, because due to its helix, 2 grooves are formed, major groove and minor groove, and 10 base pairs are present between 2 consecutive major grooves. Okay, this structure you already highlighted in this one, right? This is what is my second last slide. I have just shown the 3D structure of DNA, how it is formed and what are the rules which are forming. Again we will discuss in our next lecture, right? In 3D you can see here, this is the major groove, this is the minor groove, right? It will be somehow appears like that. And if you talk about the quaternary structure, this is the quaternary structure of a DNA, that is deoxyribonucleic acid. So friends, in today's session we have simply discussed about the biochemistry of the DNA and the constituents or components of the DNA. In next lecture, we will discuss about the double helix model of DNA, the structure of DNA in detailed way, and we will also discuss about the Irwin-Chagaff rule and some of the parts of that. Till the time, I hope I have made myself very clear. You can ask any type of doubt to your respective teachers, right? Take care, stay home, stay healthy. Bye bye.